Bismillahirrahmanirrahim. Assalamu alaikum and good afternoon to all attendees. Welcome everyone to the 6th International Conference of Applied Zoology 2023. My name is Durasamin and I am an MPhil scholar from Department of Zoology, Government College, University, Faisalabad. I am the moderator of this session, which will include the talks of the fields of genetics and histology. So without any delay, let's begin our fourth session with our first speaker, Zainab Qasim from Government College, University, Faisalabad. She will deliver a presentation on the topic titled Iron Oxide Nanoparticles of Moringa oleifera Improve Iron Deficiency Anemia in Rat Model. Please, Zainab Qasim.
Hello everyone, my name is Zainab Qasim from the Department of Nutritional Sciences at Government College University, Faisalabad. First of all, I would like to thank ICAZ-2023 for selecting my abstract to represent at this forum of 6th International Conference on Applied Zoology. My topic for representation is iron oxide nanoparticles of Moringa oleifera improve iron deficiency anemia in rat model. So here we go. Here is the abstract of my research study, and the next one is about introduction. Anemia is defined as a decrease in the circulating red blood cell mass to below age-specific and gender-specific limits. It can be caused by a variety of systemic disorders and diseases, as well as primary hematological disorders. Further, in Pakistan, iron deficiency anemia were more common among females than males. 82% patients were females and 18% were males. The major risk factors were pregnancy that contributed as 57%, nutritional inadequacy as 36%, and others are 7%. As iron deficiency anemia can be treated by many methods like giving medicines and supplements, but they have many side effects, so phytomedicines are believed to have benefits over conventional drugs and are regaining interest in current research. In this way, Moringa oleifera is a multi-purpose herbal plant used as human food and an alternative for medicinal purposes worldwide. We use Moringa oleifera leaves because they contain more vitamin A than carrots, more calcium than milk, more iron than spinach, more vitamin C than oranges, and more potassium than bananas. Next shows the different mineral composition in Moringa dried leaves, but the drawback of using these leaves is that their availability is not much higher at cell level, so different technology is used to increase their bioavailability. We prepared iron oxide nanoparticles by nanotechnology method that deals with various structure of matter having dimensions of the order of a billionth of a meter. Green approach is used for synthesis for its effectiveness, non-hazard and eco-friendly manner. Iron oxide nanoparticles in nanotechnology is a particle that defined as a small object that behave as a whole unit in terms of its transport and properties. Recently, iron oxide nanoparticles have attracted much consideration due to their unique properties such as supramagnetism, surface to volume ratio, greater surface area, and easy separation methodology. Next, the part we are going to discuss is about materials and methods. At the very first step, Moringa oleifera leaves were collected and dried under room temperature. The next step was the preparation of extract. The third step was the formation of iron oxide nanoparticles, and in last, characterization of nanoparticles was done by X-ray diffraction method, scanning electron microscope, and Fourier transform infrared spectroscopy. Here are the detailed explanation of one by one steps of a preparation of iron oxide nanoparticles. First step is the extraction, in which leaf extract was prepared by boiling at 100 degrees centigrade for 1 hour. The next step was filtration, in which the prepared extract filtered to remove impurities. Third one is mixing of solution. In this step, filtered solution mixed with iron salt solution. And the final step is centrifugation. Centrifuge the solution for solid state. In the last, we discuss about results. First, we discuss about X-ray diffraction analysis. The prepared nanoparticles were characterized by in vitro to check morphology, particle size, crystallinity, and ultraviolet visible absorption. X-ray diffraction analysis is performed to verify the nature of material prepared. It tells us whether the sample is in crystalline form or in any other phase. In the graph, the different peaks shows the crystallinity of nanoparticles. Next one is about scanning electron microscope SEM analysis. SEM analysis is used to determine the particle size of iron oxide nanoparticles. SEM images were obtained at different scales like 5 micrometer, 3 micrometer, 1 micrometer, and 0.8 micrometer. FTIR analysis is performed to check the presence of various kinds of functional groups, which exist in composites coming through different sources. Here the peaks at different values represents the presence of different functional groups. In vitro results, the last one is UV visible absorption. UV is one of the most important techniques for the characterization as it gives results about the optical studies of things. These two graphs show the absorption of iron oxide nanoparticles. In vivo studies we performed to evaluate the efficacy of the prepared nanoparticles in treating iron deficient anemic rats. First of all, we discuss about the hematological properties. Rats were distributed in 5 groups as G1, G2, G3, G4, and G5. G1 represents the negative control group, G2 represents the positive control group, G3 represents the Moringa oleifera leaves powder treatment, G4 represents the iron oxide nanoparticles treatment group, and G5 represents the drug treatment group. In all biological parameters, the lowest values are in G2 group due to anemia induction, and the highest values are in G4 iron oxide nanoparticle treatment group as compared to other treatment groups. We also performed safety evaluation tests like liver function test and renal function test to check the toxicity effect of iron oxide nanoparticles. But as we can see that there is no significant variations in all groups. This shows that the use of iron oxide nanoparticles for the management of iron deficiency anemia leaves no harmful effect on liver and kidney. This study evaluates the use of iron oxide nanoparticles for the administration of iron deficiency anemia. The net consequences of the research were found positive about the use of Moringa oleifera based iron oxide nanoparticles. These treatment method recognized as advantageous effect on the management of iron deficiency anemia and complete blood count. Thank you for your attention.
Thank you, Zainab Qasim, for sharing your expertise with us today. Our next presenter is Ikra Khalid from University of Agriculture, Faisalabad. She will deliver a presentation on the topic titled Growth Performance and Survival Rate of Euryhaline Fish Oreochromis niloticus under the influence of different salinity level. Please, Ikra Khalid.
Assalamu alaikum. I am Ikra Khalid and my topic is growth performance and survival rate of Eurohaline fish Oreochromis niloticus under the influence of different salinity levels. Let's start with the introduction. As Pakistan has many saline, aquatic, and land resources, but public thinks that these resources are useless. In fact, we can use them to utilize the different fish culturing. For example, Eurohaline fish that we can produce in saline water. I conduct this study to enhance our aquaculture in Pakistan and for the production of fish industry such as Nile tilapia. And as everyone knows that aquaculture is one of the world's largest food production. sector that resolves many problems nowadays in underdeveloped countries, for example, poverty, malnutrition, etc. But in this study, I will discuss about how climate change affect on the production of nice tilapia because it can survive in a saline water. As we know that saline water is due to the climate change, and I discussed about all the climate change reasons thoroughly. And other is the production of nice tilapia is ideal for the saline water, but why? As there are many different species, but no one can survive in the saline water, in the hypersaline water, you can say that. But nice tilapia can survive in the saline water, but why? And other thing is that effect of saline water on the growth of nice tilapia. In this study, I observed that saline that's how many ways saline water affects on the growth of nice tilapia in for example in growth rate in their survival rate. And the next is a objective evaluation of growth performance and survival rate of Nile tilapia. I evaluate the growth rate and the survival rate. And the survival rate it means how many fishes are survived at the end of the trial and optimum salinity level for better growth survival rate of Nile tilapia. As I will discuss in material and method in which I use different salinity in different salinity tanks, but one is the optimum level that is best for their growth. Next, review of literature. In review of literature, I thoroughly studied different research papers that study has been done in the past. For example, a role of aquaculture in the environmental sustainability, in which I discuss all the aquaculture things and environmental and how it can maintain the environment, water resources of Pakistan, impact of climate change on aquaculture, because this study has a huge impact on the climate change and impact of environmental sector. For example, temperature changes, pressure in water, and effect of salinity on living organisms and Nile tilapia. And as the last is biology and behavior of Nile tilapia, because this one is our model organism in which I conducted this trial. And other is material and method. In material and method, I bought fish fingerlings from the hatchery and used 5 water tanks with a different salinity level. First one is the control group, other one is with the 3.8 ppt, 4.8 salinity level, 5.8, 6.8 ppt. These are the different salinity level. And is here I use salt and salinimeter. With the salinimeter, I check the salt in water. Then I sub them feed in 2 times daily. And biometer, pH meter, reactors are used for their physiochemical meters for their better life performance. And next one is analysis. How I find these analysis and how I find the results. So I use these formulas: weight gain is equal to final weight gain minus initial weight gain, and multiply by 100. I use these formulas to get their weight gain, length gain, and survival growth rates. And after this analysis, I used Minitab software for the statistical analysis, in which I used ANOVA, one-way ANOVA variance, and the Tucky test for the comparison of the different means of the data. And in which way I found out the all results of the trial. Analysis of variance means of weight in this is the treatments in that I found out by the Innova in one it is just a model slide to see you how how the means and numbers of fishes, and you can say that it's a ANOVA structure. It's also the analysis of variance of weight gain, treatments, errors, and total, and SSMS and p-value. Most important thing is a result, in the result a highly significant difference was observed in this trial. The result is I found out the maximum growth rate and maximum survival rate in the T4, then T3, then T2, then T1 and control group. The main reason is that in T4, NaCl concentration is higher. The NaCl concentration higher, the osmoregulation and metabolism rate and the temperature rate. The higher osmoregulation and metabolism rate increases their appetite and in which way they consume more feed as compared to the different tanks. That's why their growth and survival rate is higher in T4 as compared to other treatments. So the maximum weight gain and length achieved in T4 and minimum in T2 because in T0 we not used any salt. That's why their osmoregulation and their temperature and their metabolism rate is lower as compared to T4. In the discussion, the result of my studies are the present studies are supported by the different scientists. For example, McHugh and Alton and Sarian, but one table is contradicted my studies because they use different approaches in their studies. In challenges that we should not be ignored in this trial because if we use long-term hypersaline suggests that can cause a negative side effect on the tilapia. If because 4.8 is a best optimum best level for their survival growth. If I will use 6.8, 7.8, it will cause the negative effect on their growth and their survival days. That's why it can also cause a neuroendocrine responses which can effect on their growth rate, swimming capacity, and immunity and the reproduction stage. And thank you so much for the attention.
Thank you, Ikra Khalid. Your presentation was truly impressive. Our next presenter is Mohsin Raza from University of Gujarat. He will deliver a presentation on the topic titled Relative Abundance of Beetle Fauna Among Oat Fields. Please, Mohsin Raza.
Assalamu alaikum everyone. So it's a 6th International Conference on Applied Zoology. So I'm going to discuss my topic about that international conference, and that is about the relative abundance of beetle fauna among odd fields. And I'm going to introduce my topic first. So beetles are the member of order Coleoptera, including a vast diversity of insects. The order Coleoptera contains the largest number of described species of any insect order, and they are mainly found in nearly all natural habitats, including freshwater and marine habitat. And you know, the beetles were discovered in North America in 1962 in Michigan, USA. The biology and feed dynamics of the cereal leaf beetle have been well studied and documented across different regions in the world, including southern Alberta. In general, the beetles were active from mid-March to the July, and the peak oviposition occurs in the late March to early April when succulent hosts were available. And I'm just presenting this topic as for the PhD student from the University of Gujarat, and the abstract review of my research was that oat is containing high nutrition power for livestock. So being it's a high nutritionist food content, the beetles have a very enhanced effect on the odd fields and the other nutrition related fields. And the beetles that related from the order Coleoptera is an invasive Eurasian pest of cereals including wheat, oats and barley, and expanded its geographical range significantly. So the present research work was about the to check the relative abundance of beetle fauna among odd fields. So I have done this research from the University of Agriculture, Faistabad, and about 234 specimens were recorded during the whole research study, and it was about the the abundance was that of the Cosilana septempunculata, data, that is about 53.85%, and it is nearly about 126 in abundance. It's about 25.64% in number, and that distribution in a habitat can provide insight view for the conservation efforts. And that was, that field was mainly, considered as the, the highly grouped by the, or highly, you can say, the highly expanded order Coleoptera, that is beetles were the, that was mainly present over there. So what was the objective of my findings? That keeping in view the finding of these research, the present research was designed to explore the relative abundance of beetle fauna among ords at main campus of agronomy fields from the University of Agriculture, Faisalabad. And I have chosen the cultivated area from the agronomy farm, University of Agriculture, Faisalabad, and that was majorly export crops, including the kharif crops, which include maize, rice, sugarcane, and bajra. And the research area was covered with many kinds of crops, including oat fields. I have just done the collection and identification of data from the oat fields as a sample, and a specimen were collected by selecting an area of 100 meters square of the field by following methods: by hand picking method, by using sweep net, and by using forceps. And that was stored in jars containing 70 ratio into 30% alcohol and glycerine solution, and then shifted to pest control laboratory at Department of Zoology and Fisheries at University of Agriculture, Faisalabad. Then how we can identify that the beetles are mainly over there? The collected samples were identified and stored with the aid of naked eye, magnifying glass, and under the microscope vision. All the observed specimens were arranged in the table form according to their morphological characteristics. I have done this statistical analysis by putting on the T and Z test on it to determine the various aspects of diversity from the Shannon Diversity Index were used. And what was the conclusion? I have just concluded that it has been concluded from the overall studies that owing to simpler nature, except few cases, oat fields were recorded with similar communities. All the populations were existing homogeneously and approximately experienced same number of classification representatives. And it was total 234 specimens were recorded during the whole research study, and maximum relative abundance from oat fields were according from Coccinella septim percutata, that is 53.85%, and that family was about the Coccinellidae family that was recorded in the similar contest. Thank you so much for your attention, and I hope you got the main points from my findings. Thank you so much, everyone.
Thank you, Mohsin Raza, for an amazing presentation. Our next presenter is Huma Noshin from University of Education. She will deliver her presentation on the topic titled Serological Studies in Diabetic Mice Treated with Mixed Extract of Chicory, Chicorium intybus, and Kalonji Nigella sativa seeds. Please, Huma Noshin.
Assalamu alaikum everyone. First of all, I would like to thank you, the organizing committee of ICAZ-2023, for giving me this opportunity to share my research work with scientific community. My name is Huma Noshin and I am from University of Education, Lahore. Today I will talk about serological studies in diabetic mice treated with this mixed extract of chicory and kalonji seeds. Here is the abstract of this study. If we take the diabetes into account, it is a condition which is characterized by persistent hyperglycemia, abnormal functioning of the insulin, and difficulty in the metabolism of carbohydrates, fats, and proteins. And this all occurs due to the destruction of the pancreatic beta cells. Depending upon the destruction of the pancreatic beta cells, diabetes has been divided into three types: type 1, type 2, and type 3. Type 1 is also known as autoimmune disease because in this the body's own cells destroy the pancreatic beta cells and the body totally becomes insulin dependent, and it is also known as insulin dependent diabetes. Type 2, in this there is partial degradation of the pancreatic beta cells and body produces less amount of insulin, but that is not enough for the proper functioning of the body, and that's why this is also known as non-insulin dependent diabetes. Type 3 is the gestational diabetes that occurs during the pregnancy but it may leave after the delivery of the newborn. Globally, diabetes has become a problem which affects many people. It is estimated that 400 million have been 4 million people have been affected by this disease, and it is expected that it may exceed to 600 million by 2050. According to Diabetes Prevalence Survey of Pakistan, the adult population of Pakistan is at more risk of diabetes type 2, which is 21.9 million. Approximately out of 463 million diabetic people, greater than 90% of the people have type 2 diabetes living in the neighboring countries of Pakistan that are India and China. According to recent International Diabetes Federation, 33 million people in Pakistan are suffering from diabetes, and this country has the third largest diabetic population. Diabetes mellitus can give rise to alarming effects on different systems of the body, such as renal functions, heart, liver, and vascular system. Diabetes kidney disease is a common and morbid complication of type 2 diabetes, which affects 40% of the patients. Individuals with type 2 diabetes have higher incidence of liver function test abnormalities, and it is thought that elevations of transaminases reflect underlying insulin resistance. Occasionally, type 2 diabetes has been thought to be the risk factor of liver diseases, and it has become the third largest killer of the mankind after the cancer. Though a large number of conventional drugs having anti-diabetic potential, but they pose serious side effects. Standard treatment is failing to achieve of the blood, glucose, kidney, and liver functions in many patients. The use of modern medicine to treat the diabetes has been reduced due to their side effects. Many medicinal plants have recently attracted attention due to their important role as dietary supplements with health promotive effects and minimal side impacts. Herbal treatments are more popular because of their less negative consequences, readily availability, and less price. There are very benefits of medicinal plants, including chicory antibus and Nigella sativa are one of them. Biological activities and therapeutical uses of Nigella sativa and chicory antibus are widely studied in the present times. Both of them are health promotive and disease preventive. Chicory and its products have been used to treat a range of diseases, and it is also used as complementary and alternative medicine to reduce liver and gastrointestinal disorders and also diabetes. Taking into account these therapeutic effects of chicory and kalwanji, this study is also based on the use of these two plants, and the problem statement of this study is serological studies in diabetic mice treated with mixed extract of chicory and kalwanji seeds. Objectives of this study were set to prepare mixed extract of kalwanji and chicory seeds, to determine their anti-diabetic potential by using their mixed aqueous extract in diabetic mice, and to study the serology of liver and kidney in diabetic mice treated with mixed extract of N sativa and intibus. This is the methodology. First step is the preparation of the plant extract, then animal handling, experimental design, application of the treatment, and at the last, sample collection. First of all, preparation of the plant extract. For the preparation of the plant extract, the seeds of the C. intibus and N. sativa were collected and cleaned. The seeds of both plants were ground and separately and stored. The seeds of C. intibus and N. sativa were collected and washed thoroughly. Aqueous extract was prepared by using the powdered seeds and was prepared with distilled water using 3:1 quantity of seeds of kalwanji and chicory respectively. After they were kept overnight, soaked, and then on the next day they were boiled for 15 minutes. Then it was cooled at room temperature, and then it was filtered by using the filter paper, and then final extract was ready to use. For the further process, the animals that were mice, male albino mice, they were acclimatized for 2 weeks. They were fed with a normal basal diet, and water and feed were given at labitium, and they were provided with a 12-hour light or dark cycle, and they have free access to food and water and oxygen. After the acclimatization, the mice were injected with the alloxane monohydrate injection at the dose of 150 mg per kg for the induction of diabetes. On the 7th day, the confirmation of the induction of diabetes was done, and after the confirmation of these, the experimental design was designed. The animals were divided into two groups: main groups, control group, and the treatment group. Control group was further divided into positive control group and negative control group. Positive control group was treated with standard drug that was market available, that is metformin, glucophage. And negative control group was further divided into three groups. One was treated with casni seed extract, one was treated with black seed extract, and one was treated untreated, one was left untreated because it was left for natural recovery. The next one is the treatment group. Treatment group was divided into four groups, and it was treated with mixed aqueous extract at different doses. One dose was 300, one was 400, and one dose was 500 mg per kg, and the fourth group was the paste that was prepared by using kalwanji and kasni seed. And one group was the baseline group, that was normal healthy mice. They were fed on normal diet. After the experimental design, the treatment was given to the mice for 28 days, once a day. And after the completion of the trial of 28 days, the blood sample was taken on 29th day of the treatment by direct heart puncturing. The mice were made unconscious by using chloroform, and the blood was drawn by using a syringe. Then the blood was collected in the serum gel tubes for separation. After the collection of the samples were analyzed for serological studies of liver and kidney. Blood was analyzed for serum bilirubin, alanine aminotransferases, aspartate aminotransferases, alkaline phosphatases, total proteins, and bilirubin. For kidney, urea and creatinine level were analyzed. After that, the statistical analysis were results of the study showed that when the alloxane monohydrate was introduced and diabetes was induced in the mice, the level of the glucose was very high. It got elevated, but when the treatment was applied gradually after 4 weeks, the glucose level was normalized, and the effective decrease in the glucose level was seen in the 4th week of the treatment. The same effect was seen on the body weight after the alloxane injection. Within a week, the body weight of the mice reduced, while when they were treated with the mixed aqueous extract at different doses and paste of Nigella sativa and Chicoria mentibus. At the end of the treatment, their body mass was also normalized. The effect of the treatment on the liver enzymes was also analyzed. It was analyzed that there was significant difference in the values of the ALT, ALP, and total proteins, while there was also significant difference in the albumin level, and there was no difference found in AG ratio. If we take into account the creatinine and urea, in urea there was a significant difference found, while in case of creatinine no difference was found. And it was also observed that at the dose of 400 mg per kg there were more significant results and more effective results were seen. Conclusion: in the current study, the significant difference was found in some parameters of liver, including ALT, AST, ALP, total proteins, and albumin. While in case of kidney, there was significant difference found in the creatinine level, and at the dose of 400 mg per kg of mixed extract was found to be more effective to normalize the blood glucose level, LFTs and RFTs, including ALT, AST, total proteins, creatinine as compared to normal control group. While the dose of mixed extract at the 300 mg per kg and paste were found to show the normal results of total proteins and albumin that were in line with the normal control group. That was all about my work. Thank you so much for your attention. Allah Hafiz.
Our next presenter is Alishba Arooj from University of Agriculture, Dera Ismail Khan. She will deliver a presentation on the topic titled Investigation of Intestinal Parasitic Prevalence Across Different Territories of District Dera Ismail Khan. Please, Alishba Arooj.
Assalamu alaikum. First of all, I would like to thank the organizing committee of ICAZ-2023 for providing this opportunity to share my research study. I am Alishba Arooj from the University of Agriculture. I am here to present my research study with title Investigation of Intestinal Parasitic Prevalence in Hen across different territories of District Dera Khan under the supervision of Dr. Inatul Rahman with the guidelines of Dr. Shoaib Alam, Imranullah and Masuma Batul. The table of my content include introduction, objective, material and method, result and conclusion. I would like to start from introduction. Chicken represents one of the most widespread farm animal. Chicken meat represents an important source of animal protein and fat as well as the source of organic and inorganic substance. The domestication of chicken started approximately 5,000 years ago. Since that time, chicken, egg and meat become a common source of animal protein for humans. Taxonomical classification of chicken is Kingdom Animalia, phylum Chordata, class Aves, genus Gallus, scientific name Gallus domesticus. Parasite, an organism that lives in or another organism and gain an advantage at the expense of that organism. Parasites are most frequently found in gastrointestinal tract. Two types of parasite, internal parasites and external parasites. Internal parasites contain nematodes and gastrodes. My research study related to internal parasite, that is endoparasite infestation. Intestinal parasites can pose health implications for hens such as impaired weight gain, growth, decreased egg production, increased mortality and possibly anemia. According to Perman et al. 1999, Ascarida galli approximately 64% and 73% of organic hens. Clinical signs of parasite infestation include unfitness, poor growth, feed conversion, decreased egg production, and cause death in severe cases. Pathogenicity can vary and depending on the number of worms present in hen. For example, retarded growth rate, reduced appetite, decreased egg production, blood stain, and cause weight loss by Small 1996. Keeping the view the above information, my research work is designed to carry out following objectives. That is, detection of tapeworm, Retina echinobotrida, and roundworm Ascarida galli in hens of District Dera Ismail Khan, and determine the prevalence of tapeworm and roundworm in hens of District Dera Ismail Khan. Material and methodology. First, design the study. During the period of March to April 2022, fecal samples are collected from different areas of District Dera Khan. Then that fecal samples are brought to lab of the zoology department for further procedure. The methodology start with different steps. Step 1 is intestine separation. First, the intestine separate from the body. Then fecal samples are placed in the petri dish and dissolved with distilled water for filtration. Step 2 is filtration. Filtration is the process that is used to separate the solid from the liquid by using filter paper. After filtration, centrifuge that fecal sample. Centrifugation is the technique that is used to separate the solid material from the solution according to the shape, size and density. The top filtrate is then centrifuged at 400 rpm at 3 minutes. After centrifugation, slide prepared through the direct seminar. After the slide prepared, slides are placed in the light microscope using 10x and 40x objective lens. The eggs and larvae of the tapeworm and roundworm were detected. For data analysis, prevalence is equal to number of positive cases divided by number of total sample multiplied 100. Results. About 250 samples of hen were examined for the detection of endoparasite from different area-wise sample collection From city DI Khan 30 samples were collected, from Harpur 60 were collected, from Rabin 80 samples were collected, and from Dhaki 80 samples were collected. Prevalence of Echinobothida in area of different areas of district DI Khan. Out of 30 samples from DI Khan, 3 were positive, that is 10%, while in Harpur 5 samples of Echinobothida were positive, that is 8.3% prevalence. Similarly, in Draben, 9 were positive, that is 11.5% prevalence. From Dhaki, out of 80 samples, 10 were positive, that is 12.5% prevalence. Prevalence of Echinobothida in Dhaki is high, that is 12.5%, and low in city DI Khan, that is 10% prevalence rate. Prevalence of Ascarida galli in area of DI Khan. About 250 samples for Ascarida galli was taken. From Harpur, 60 samples were taken, in which 3 were positive, that is 5% prevalence. From city DI Khan, 30 samples were collected, in which 1 were positive, that is 3.3% prevalence. Similarly, 80 samples were collected from Draban, in which 7 were positive, that is 8.75 prevalence. From Dhaki, 9 were positive out of 80, that is 11.25% prevalence. Prevalence rate of Ascarida galli is high in Dhaki with prevalence rate 11.5 and low in DI Khan city with prevalence rate 3.3. The conclusion of my research study demonstrated that Echinobothida is omnipresent parasitic intestinal disease. It could strongly decrease the production performance in domesticated chicken in different areas of DI Khan. The finding of my research study indicate that contaminated food is the major source of infection. My research study concluded that Echinobothida and Ascarida galli are the endoparasites that cause infection in chicken of Dera Ismail Khan. Echinobothida with high prevalence rate, that is 12%. The most prevalent causative agent of Echinobothida among the parasitic species was polluted oocysts that cause heavy economic losses to the poultry industry. Thank you for your attention.
Our next presenter is Momina Khan from Women University, Multan. She will deliver a present topic titled Toxicity Assessment of Carbon, Gold, and Silver Nanoparticles on Earthworms. Please, Momina Khan.
Bismillahirrahmanirrahim. The topic is Toxicity Assessment of Carbon, Gold and Silver Nanoparticles on Earthworm. यानी हमने नैनो पार्टिकल्स कार्बन, गोल्ड और सिल्वर का टॉक्सिसिटी की असेसमेंट करनी है अर्थवॉर्म्स के ऊपर. These are my total abstract introduction, then experimental objective synthesis, toxicology analysis, results and conclusions. Nanoparticles are an essential part of life. They are very beneficial to mankind, but on the other hand, they are harmful and toxic for the entire environment. The toxicity of nanoparticles increase to the lithosphere. As a result, soil organisms are disturbed, causing them not to function properly for soil fertility. जैसे कि हम सबको पता है कि earthworm जो है is the detritivorous and lives only in soil, and nanoparticles have very harmful effects on soil, and as a result it indirectly damages the earthworm. In this research, nanoparticles were synthesized. Carbon nanoparticles were synthesized from 3 different sources: brick kiln, carbon, charcoal and green tea waste. Silver and gold nanoparticles were synthesized from jettison. Their toxicity tests are conducted on earthworms. Earthworms were exposed to different concentration of carbon and silver and gold nanoparticles on filter paper for 48 hours. The brick kiln carbon induced slight toxicity. Even the lethal dose could not determine. The lethal dose of carbon, charcoal, and green tea waste nanoparticles after 48 hours of exposures was 0.4 milligram meter. And this study was used to determine the toxic effects of different concentration of carbon, silver, and gold nanoparticles on earthworm. The results showed that these nanoparticles were very toxic to earthworm. The keywords which I used in this chapter is nanoparticles, carbon, silver, gold, earthworms, reactive oxygen species, DNA damage, and comet assay. Introduction में I will use to say several types of nanoparticles such as carbon nanoparticles have widely application in industrial, agriculture, medical, and environmental sections with consumer market that increase day by day. Soil microinvertebrates include a variety of organisms such as earthworm, insects, isopods, and higher concentration of CNPs cause total reproductive failure, cause toxic effect by including oxidative stress and inflammatory reactions. Objective to determine the toxic effect of different concentration of gold, silver, and carbon nanoparticles in earthworm. Synthesis from brick kiln soot, synthesis from charcoal carbon, synthesis from green tea waste, synthesis of silver nanoparticles, and synthesis of gold nanoparticles. First of all, this is the summary to overall kiln soot from brick kiln plus 1 meter nitric acid plus absolute ethanol 10 times with deionized water, and then adjust the pH at 7.0. Then heat it at 30 degrees centigrade for 4 hours, and then centrifuge at 3000 revolutions per minute for 40 minutes. The fine carbon charcoal nanoparticles were synthesized. Collect the green tea waste, air dried, carbonized at 400 degrees centigrade for 1 hour using mortar. Potassium hydroxide was mixed with GTVA. Stirring it 4 hours with 1 molar HCl to eliminate the potassium hydroxide. The residue was filtered, dried at 120 degrees centigrade, 12 hours. GTWCNP prepared. Synthesis of silver nanoparticles: 0.5 gram chitosan plus acetic acid plus 0.5 gram silver nitrate dissolved in water. 5 ml chitosan plus 5 ml silver nitrate solution autoclaved at 120 degrees centigrade for 3 hours at 15 psi. CCNPs are prepared. And synthesis of gold nanoparticles: chitosan plus 1% acetic acid solution, 3 ml chitosan solution plus 1.5 ml, heating and stirring for 15 minutes at 55 degrees centigrade. Color change indicates the product. Toxicogenic analysis of soil microfauna earthworm. Filter paper test. Add synthesized CNPs on filter paper. Take 9 cm petri dishes, set filter paper. Add 2 ml acetone, re-moisturize filter paper, and add 2 ml distilled water. On 1 earthworm, put in 1 petri dish, kept at 20 degrees centigrade for 48 hours. Observe the survival rate. These are the overall results. The concentration of nanoparticles were ranges from these values. Conditions was also same. Observe earthworms after 7 hours, 24 hours, and 48 hours. Heat the soil for 1 hour at 150 degrees centigrade. Concentration is 210 and 50 milligram per kg of soil. Glass jars for experiment. 3 glass jars contain 500 milligram soil and nanoparticles. Each jar contains 10 earthworms. Cover the jar with fresh wrap for ventilation. Made small hole, kept in 20 degrees centigrade for 10 days. And these are the overall filter paper contact test of CCNPs. The results of nanoparticles were highly toxic and no worm survived before 24 hours. The lethal dose of CC and Ps and these values are respectively. The results of above experiment shows that PKC and Ps show slightly toxicity than CC and Ps because only BKC and Ps exposed earthworms were survival after 48 hours of exposure in filter paper contact test. The lethal dose of CC and Ps and these value, the results of soil test gold show negative results because the number of earthworms died was only 6. Jetison silver nanoparticles are toxic even in low amount because the size and shape affect sharply. Overall, the carbon nanoparticles appeared acutely toxic only when their dose was greatly than their environmentally equivalent concentration, and the nanomaterial type was essential in indicating the acute toxicity. And these are the references. Thank you so much.
Thank you, Momina Khan, for an amazing presentation. Our next presenter is Sayyida Nadia Ahmed from University of Chakwal. She will deliver a presentation on the topic titled Ameliorative Potential of Olea ferruginea fruit pulp extract against CCL4 induced testicular histopathology in mice. Please, Sayyida Nadia Ahmed.
Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to all participants in 6th International Conference on Applied Zoology. I'm Dr. Sayyida Nadia Ahmed from Department of Zoology, University of Chakwal. And the topic of my presentation is the ameliorative potential of Olea ferruginea fruit pulp extract against CCL4 induced testicular histopathology in mice. The aim of the study was to investigate the ameliorative capacity of wild olive Olea ferruginea fruit pulp extract and to suggest its possible enteroregenerative process against carbon tetrachloride intoxicated mice testicular damage. The industrial shape of seminiferous tubule with proper spermatogenic cell and the lumen was filled with the healthy sperms. While between the seminiferous tubule, a healthy interstitial tissue was found with the Leydig cell. While in the section B, you can see here that in CCL4 treated group, the shape of seminiferous tubule was disturbed and sperm head that were in dislodged form were seen here and there in the seminiferous tubule, and a proper symmetry and alignment of spermatogenic cell was totally disturbed. And there was no lumen, and if lumen is found that is too much wider, that shows totally destruction of spermatogenesis and spermiogenesis and dead spermatozoa. While in third group, that is the Olea ferruginea group, here a little bit rehabilitation is seen because seminiferous tubules have some spermatogenic cell that are in healthy condition, and lumen shows a proper cluster of spermatogenic cell and healthy sperm spermatozoa, and between the seminiferous tubule, interstitial tissue was healthy with maximum number of Leydig cells. The picture is very clear in the figure 2. Here A section shows normal seminiferous tubule with multiple and proper aligned spermatogenic cell, and lumen is filled with the healthy spermatozoa. While in B section, that is with treated CCL4 group, there is no proper symmetry and alignment of spermatogenic cell, and here you can see clearly the dislodged sperm's head that are found in the random positions. While in Olea ferruginea treated group, a proper symmetry of spermatogenic cell is found with a cluster of spermatozoa in the lumen of the seminiferous tubule. The micrometric parameters include CSA, cross-sectional area of seminiferous tubule, CSA of spermatogonia, CSA of spermatocytes, head of spermatozoa, and tail length of sperms. The conclusion is that best enteroprotective fruit is Olea ferruginea, and the result shows that the medicinal wild fruit shows excellent nutraceutical potential against enteropathologies created by CCL4 exposure. Thank you so much for your attention.
Thank you, Sayyida Nadia Ahmed, for sharing your amazing research work. Topic titled Risk Factors of Diabetic Retinopathy Patients of Type 1 Diabetes Mellitus in Hyderabad and Jamshoro. Please, Dr. Fahmida Channa.
Bismillahirrahmanirrahim. Assalamu alaikum. This is Dr. Fahmida Channa. Today I am going to presenting my research work on risk factors of diabetic retinopathy patients of type 1 diabetes mellitus in Hyderabad and Jamshoro. Content of presentation. First of all, I would like to share the introduction of diabetes mellitus. Diabetes mellitus is a major health problem globally. Diabetes mellitus is an endocrine disorder due to deficient secretion of insulin or improper action of insulin leads to be hyperglycemia. Globally, the prevalence of diabetes mellitus in 2014 was 8.5% among the adult population. This figure shows that type 1 diabetes mellitus. Type 1 diabetes mellitus is autoimmune disorder in which the beta cells of islets of pancreas are destroyed. Therefore, no insulin secretion, and least insulin secretion leads to be the high glucose level in the blood, and increased level of glucose in the blood is said to be hyperglycemia. Prolonged hyperglycemia lead the microvascular complication, which is the most common microvascular complication, that's said to be the diabetic retinopathy. And the diabetic retinopathy prevalence worldwide is about 34.6%. Diabetic retinopathy is a well-known sight-threatening microvascular complication of diabetes mellitus. The prevalence of diabetic retinopathy in well-developed nations has been thoroughly investigated and the risk factors are well understood. This figure shows the risk factors of diabetic retinopathy, age groups, hypertension, degree of glycemic control, duration of disease, smoking, genetic factors. To talk about the aim and objectives of my research work, to assess the diabetic retinopathy risk factors, just like a duration, gender, family history, smoking, and blood pressure among the type 1 diabetic patients on follow-up at hospitals of Hyderabad and Jamshoro, Sindh. To observe the frequency of clinical manifestation of diabetic retinopathy patients due to risk factors. Now, discuss about the material and method of research work. This study was an institution-based cross-sectional study. Data collection was conducted from month of July to December 2018 during routine working days. Data were collected through self-developed questionnaire in English version. Patients of different ages of both sexes were also recorded with their controlled or uncontrolled blood pressure. Diabetic based on blood glucose measurement, either random or fasting. Questionnaire related to the age group or duration of the patients were designed and graphically explained here. This tabular form shows the questionnaire about risk factors of diabetic retinopathy in the type 1 diabetes mellitus. Results. This tabular form is showing the gender-wise distribution of diabetic retinopathy patients. 66.6% were male, 33.3% of female. This figure shows the 55.5% of patients were the age group 26 to 30 years, 44.4% of patients were the age group 20 to 25 years. This table shows the duration of the period diabetic retinopathy patients. About 55.5% of patients had the duration were 50 to 20 years. 33.3% of patients the duration of diabetes were 10 to 15 years, 11.1% of patients for the duration of disease up to 10 years. This figure shows the family history of diabetic retinopathy patients were 66.6% of patients were the negative family history, and the 33.3% of patients were the positive family history. This figure shows the blood pressure status of diabetic retinopathy patients. About 55% of patients the hypertensive, and the remaining 44% of patients were the normotensive. This figure shows the glycemic status of diabetic retinopathy patient, and that risk factors, the most common risk factors which leads to be the diabetic retinopathy in the type 1 diabetes mellitus. That shows 83% of patients were the uncontrolled glycemic status, and the remaining 17% of patients were controlled glycemic status. In the last, I will describe the conclusion and recommendations of my research work. Prevalence of diabetic retinopathy was high, about 44%. More than half of the patients who are the male, they have the diabetic retinopathy. More than half of the DR patients, duration of type 1 diabetes mellitus 15 to 20 years. Major risk factors of diabetic retinopathy of type 1 diabetes mellitus patients are gender, duration of disease, age group, negative family history, glycemic uncontrolled status, and hypertension were found to have significantly associated with diabetic retinopathy. Now the recommendation. The results emphasize that the Ministry of Health should establish strategies and policies to control diabetic retinopathy. We recommend that health workers also provide sustainable health information to diabetic patients on possible risk factors of diabetic retinopathy, just like hypertension, duration of diabetes, negative family history, and poor glycemic control, as they were evidenced in the current study. Acknowledgement. At first, I pay very deepest gratitude to Almighty Allah who honored me to potential and existing ocean. I wish to thank the team of organization of 6th International Conference on Applied Zoology 2023. I wish thanks to my Honorable Professor Dr. Tahira Jabin. Thank you for your attention.
Thank you, Dr. Fahmida Channa, for your valuable contribution to today's discussion. Ladies and gentlemen, as we wrap up today's presentation, let me express my gratitude for your attention. Thank you for being a wonderful audience. Thank you so much.